right, class, today we are going to begin Chapter 7, Microbial Nutrition, Ecology, and Growth. Our learning outcomes are to list the essential nutrients of a bacterial cell, differentiate between macronutrients and micronutrients, list and define 4 different terms that describe an organism's sources of carbon and energy, define saprobe and parasite and provide microbial examples of each, compare and contrast the processes of diffusion and osmosis, identify the effects of isotonic, hypotonic, and hypertonic conditions on a cell, name 2 types of passive transport and 3 types of active transport. So what is nutrition? Nutrients are acquired from the environment and used for cellular activities. An essential nutrient is any substance, whether in elemental or molecular form, that must be provided to an organism. Macronutrients are required in relatively large quantities. They play principal roles in cell structure and metabolism. Micronutrients are trace elements. They are present in smaller amounts. They are involved in enzyme function and maintenance of protein structure. Inorganic versus organic nutrients. An inorganic nutrient is an atom or a simple molecule that contains a combination of atoms other than carbon and hydrogen. Organic nutrients contain carbon and hydrogen atoms. They are usually the product of living things. So here are some principal inorganic reservoirs of elements. Carbon dioxide in the air, CO2 and CO3 in rocks and sediments, oxygen, O2, oxygen in the air, certain oxides in water, nitrogen in air, hydrogen, water, H2, gas, and mineral deposits. Phosphorus contains mineral deposits of PO3 and 4, H3PO4. Sulfur has mineral deposits. Volcanic sediments, and potassium has mineral deposits such as in the ocean. Potassium chloride, K3 and PO4. Sodium, where do we get sodium? Mineral deposits in the ocean. Sodium chloride. What is sodium chloride? Salt. Calcium, mineral deposits in the ocean. Magnesium, mineral deposits, geologic sediments. Chloride from the ocean. Iron, mineral deposits, geologic sediments. And manganese, molybdenum, cobalt, nickel, zinc, and copper. And other micronutrients, small trace elements, amounts, various geologic sediments. Uh-oh, I didn't make that one wide. It's too late now, isn't it? All right. Analysis of the chemical composition of an Escherichia coli cell. What do we already know about E. coli? Is it always good for us? Is it always bad? Sometimes good, sometimes bad. H70157 can kill you. What makes vitamin K in our stomachs? E. coli does. It's good in that sense. So the organic compounds and their percent total weight and dry weight are listed here. Proteins, DNA, RNA, carbohydrates, lipids, and other miscellaneous compounds. The inorganic compounds are water, and all water is total weight is 70%. And of course it doesn't have a dry weight because it's water. And all others have a 1% total weight. Then over here under the elements, these are compounds, these are elements. Carbon, oxygen, nitrogen, hydrogen, phosphorus, sulfur, potassium, sodium, calcium, magnesium, chlorine, iron, and trace metals. So the carbon source is for a heterotroph, an organism that must obtain its carbon in organic form. It's dependent upon other life forms because the word heterotroph itself means that it cannot make its own food, right? So it's dependent on other life forms. Most carbon sources exist in a form that's simple enough for absorption. Larger molecules must be digested by the cell before it can be absorbed. So carbon sources for the autotroph, the self-feeder, able to make its own food. It's an organism that uses inorganic carbon dioxide as its carbon source. It has the capacity to convert carbon dioxide into carbon compounds. It's not nutritionally dependent on other living things. Here are some nitrogen sources. The main reservoir is nitrogen gas. 79% of the atmosphere is indispensable to structure of proteins, DNA, RNA, and ATP. Primary nitrogen sources for heterotrophs must be degraded into basic building blocks in order to be utilized. Some bacteria and algae utilize inorganic nitrogenous nutrients. But regardless of the source, nitrogen must be converted to NH3 before it enters the cell. This is the only way. This is the only form that can be directly combined with carbon to synthesize amino acids and other compounds. What about the oxygen sources? Oxygen plays an important role in the structural and enzymatic functions of the cell. It's the major component of carbohydrates, lipids, nucleic acids, and proteins. It is a common component of inorganic salts, and oxygen makes up 20% of the atmosphere. Hydrogen sources have overlapping roles in the biochemistry of cells, including maintaining pH, forming hydrogen bonds between molecules, and serving as a source of free energy in oxidation reduction reactions of respiration. Phosphorus or phosphate sources. The main inorganic source is phosphate, derived from phosphoric acid, H3PO4. It's found in rocks and oceanic mineral deposits. It's the key component of nucleic acids, essential to genetics of cells and viruses because it is the key component of nucleic acids. It's found in ATP, which is an important energy molecule in cells. Sulfur sources are widely distributed throughout the environment in rocks and sediments. They are an essential component of some vitamins, vitamin B1 and amino acids, methionine and cysteine. They form disulfide bonds that help determine the shape and structural stability of proteins. Essential organic nutrients. They are a growth factor. An organic compound such as an amino acid, nitrogenous base, or vitamin that cannot be synthesized by an organism. An example would be the essential amino acids. They must be provided by the environment. So, how do microbes feed? The nutritional types are phototroph. These are microbes that can photosynthesize. Or chemotroph, microbes that gain energy from chemical compounds. Here are the nutritional categories of microbes by energy and carbon source. The autotrophs are listed up top. Carbon dioxide, the energy source, is the non-living environment. So photoautotrophs must use sunlight. And an example, photosynthetic organisms such as algae, plants, and cyanobacteria. We'll need to know that about the cyanobacteria when we do our lab next week. The chemoautotroph, simple inorganic chemicals, only certain bacteria or archaea such as methanogens, deep sea vent bacteria. Then for our heterotrophs, the carbon source, organic, heterotroph organic, we have the photoheterotroph, which uses sunlight as its energy. An example would be the purple and green photosynthetic bacteria. A chemoheterotroph utilizes the tissues and fluids of a live host. It's a parasite. And the chemoheterotroph metabolizes the organic matter of dead organisms, a saprobe. An example would be fungi and bacteria. So, what is the difference in a parasite and a saprobe? Thank you. Here's a photoautotroph and a chemoheterotroph. Photoautotrophs capture energy from light rays and transform it into chemical energy that can be used for cell metabolism. They produce organic molecules that can be used by themselves and heterotrophs, and they form the basis for most food webs. Chemoautotrophs, chemoorganic autotrophs use organic compounds for energy and inorganic compounds as a carbon source. We also have lithoautotrophs that require neither sunlight nor organic nutrients, and they rely totally on inorganic materials. Methanogens. What are methanogens? They are, methane gases. Chemoautotrophs that produce methane from hydrogen gas and carbon dioxide. They are formed in anaerobic, hydrogen-containing microenvironments of soil, swamps, mud, or intestines of some animals. Anaerobic. What does that mean? Without oxygen. Archaea, some of which live in extreme habitats such as ocean, vents, and hot springs up to 400 degrees Celsius. Methane can be used as a fuel and plays a role as a greenhouse gas. Chemoheterotrophs, the majority of heterotrophic organisms, they derive both carbon and energy from organic compounds. Organic molecules are processed through respiration or fermentation, release energy in the form of ATP. Aerobic respiration is the principal energy-yielding pathway in animals, protozoa, fungi, and aerobic bacteria. Aerobic means in the presence of oxygen. It's complementary to photosynthesis. Glucose and oxygen are reactants, and carbon dioxide is given off. The Earth's balance of energy and metabolic gases is dependent upon this reaction. Saprobic microorganisms are decomposers of plant litter, animal matter, and dead microbes. They are important in recycling nutrients that are held in organic materials. Most saprobes have a rigid cell wall and cannot engulf large particles of food. Examples, bacteria and fungi. They release enzymes into the environment to digest food into smaller particles that can be transported into the cell. We'll pick up next time here. Let's look at this picture first. This is the extracellular digestion in a saprobe with a cell wall. Could be a bacterium or a fungus. The wall cell is a barrier in A here. In B, enzymes are transported outside the walls. What do enzymes do? What are enzymes? They do what? Speed up, slow down, metabolic processes. In C here, these enzymes hydrolyze the bonds and nutrients. Be out in a minute. Smaller molecules are transported across the wall into the cytoplasm here in D. Okay, so we will pick up the next time with slide 15. We'll get ready to go to lab and do our peer review. Taylor.
Yes, I am.
Hit it again, right?
I'm gonna try.
Huh?
I'm gonna try. Did you stop it?
Click here to pause.
Yeah, click that. Okay. Is it still recording? Hit this minus arrow. You should not still have the screen. Okay, hit that again, see what happens.
Cross it off. I get them out. Just wait.
Where did it go?
Oh, wow.
I hope it didn't delete that.
I didn't do anything to delete it.
I know, I don't think you did. Just try to hit this little line and see what comes up. Like if you tap it, what does it do?
You need to keep going.
Hit pause.